As the world's population grows older, the pursuit of not just a longer life but a healthier one has become a central focus in biomedical research. Today, two-thirds of deaths globally are linked to age-related diseases, making healthy aging a top priority. Traditional approaches have targeted the whole body, using genetic regulation or drugs to slow aging. But recent advances are shifting the spotlight to the unique differences between individual cells. This new era, powered by single-cell sequencing, allows scientists to analyze tissues cell by cell, uncovering the hidden diversity that shapes how we age. In this video, we'll explore how these breakthroughs are transforming longevity interventions, setting the stage for more precise and effective strategies. Living longer is only part of the story. What truly matters is living healthier for longer. The concept of healthspan, the period of lifespan in good health, is now at the forefront of aging research. Scientists are investigating interventions that not only extend life, but also delay the onset of diseases like diabetes, cancer, and neurodegeneration. This shift in focus has led to a surge in studies on how to maintain vitality, independence, and well-being as we age. By understanding the mechanisms behind healthy aging, researchers hope to develop strategies that help people enjoy more active and fulfilling years, not just more years overall. For decades, scientists have explored a range of interventions to slow aging and extend life. These include dietary restriction, exercise, genetic manipulation, and pharmacological treatments. Dietary restriction, for example, has been shown to extend lifespan in species from yeast to monkeys, while exercise boosts cardiovascular and metabolic health. Genetic approaches target pathways like insulin IGF-1 and mTOR, which regulate growth and metabolism. Drugs such as metformin and rapamycin mimic some of these effects, offering new hope for age-related disease prevention. Yet these strategies often act broadly, affecting many tissues and cell types at once, which can lead to mixed results and unintended side effects. Genetic manipulation has uncovered powerful ways to influence aging. In model organisms like C. elegans, reducing the activity of the DAF2 gene, part of the insulin IGF-1 pathway, can double lifespan. Similar pathways exist in humans, where variants in the FOXO3 gene are linked to longer life. Another key player is the mTOR pathway, which controls cell growth and metabolism. Inhibiting mTOR, either genetically or with drugs like rapamycin, has been shown to extend lifespan in worms, flies, and mice. These discoveries highlight the deep evolutionary roots of aging and suggest that targeting these pathways could benefit human health span as well. Dietary restriction, or reducing nutrient intake without malnutrition, is one of the most robust ways to extend lifespan across species. It works by activating nutrient-sensing pathways like IIS, AMPK, and mTOR, which regulate metabolism and stress responses. Building on this, scientists have developed nutraceuticals, bioactive dietary compounds such as resveratrol and polyphenols, that mimic the effects of dietary restriction. These compounds enhance mitochondrial function, vascular health, and glucose metabolism, offering broad benefits for aging tissues. However, the effects can vary widely depending on the individual's genetics and the specific tissues involved. Regular physical activity is a powerful non-pharmacological intervention for healthy aging. Exercise improves cardiovascular and metabolic health, boosts neurogenesis, and reduces inflammation. At the molecular level, it triggers the release of myokines, muscle-derived factors that communicate with organs throughout the body. These molecules, including irisin and BDNF, help maintain brain health and resilience. Exercise mimics some of the beneficial effects of dietary restriction and mTOR inhibition, making it a cornerstone of any longevity strategy. Its broad, systemic benefits make it one of the most accessible and effective ways to promote health span. Drugs like metformin and rapamycin are at the forefront of pharmacological approaches to aging. Metformin, widely used for diabetes, activates AMPK and has shown promise in extending lifespan in some animal studies. Rapamycin, an mTOR inhibitor, extends lifespan in mice and is now being tested in human trials for its potential to delay aging. Other compounds, such as acarbose and senolytics, are also under investigation. While these drugs target conserved aging pathways, their effects can differ across tissues and individuals, highlighting the need for more targeted approaches. Targeting senescent cells, cells that have stopped dividing and contribute to aging, is a promising new strategy. Senolytics are drugs that selectively eliminate these cells, improving tissue function and extending lifespan in animal models. In humans, early trials with senolytics like dasatinib and quercetin have shown improvements in physical function. Another frontier is cellular reprogramming, where factors like OCT4, SOX2, and KLF4 are used to reset the cellular aging clock. Partial reprogramming can rejuvenate tissues and reverse signs of aging, but must be carefully controlled to avoid harmful side effects. Blood-based interventions such as parabiosis and plasma exchange have shown that young or reset blood can rejuvenate organs in animal models. These treatments modify the systemic signaling environment, promoting tissue regeneration and reducing inflammation. Key molecules like GDF11, TIMP2, and oxytocin have been identified as contributors to these effects. While promising, these interventions are complex and their benefits are not limited to a single pathway or cell type. Ongoing research aims to translate these findings into safe and effective therapies for humans. Despite their promise, current longevity strategies have significant limitations. Interventions that benefit some cell types may harm others. For example, rapamycin extends lifespan but can impair fertility, and genetic manipulation of insulin IGF-1 pathways may reduce growth or metabolic health. In vivo reprogramming, while rejuvenating some tissues, can cause organ failure if not carefully managed. These challenges underscore the complexity of aging and the need for more precise, cell-type, specific interventions that minimize side effects and maximize benefits. Single-cell sequencing has revolutionized aging research by enabling the analysis of tissues at unprecedented resolution. Unlike traditional methods that average signals across millions of cells, single-cell technologies reveal the unique aging patterns of individual cell types. This approach has led to the creation of aging cell atlases in model organisms like worms, flies, and mice. These atlases map gene expression changes across the lifespan, uncovering which cells age fastest and which are most vulnerable to disease. The insights gained are paving the way for more targeted and effective longevity interventions. The creation of a single-cell aging atlas in C. elegans, a tiny roundworm, has provided deep insights into how different cell types age. Researchers profiled over 47,000 somatic cells, finding that energy metabolism genes decline universally, while stress response genes vary by cell type. Neurons in particular showed the greatest transcriptomic changes, suggesting they may drive early aging in worms. The atlas also identified new longevity regulators such as J3, LSY2, and MEF2, which act in a cell-type specific manner. These findings highlight the importance of targeting specific cell populations to delay aging. The Aging Fly Cell Atlas, AFCA, offers a comprehensive view of how aging unfolds in Drosophila, the common fruit fly. The study found that muscle cells experience the greatest loss with age, mirroring sarcopenia in humans. Adipose tissues like oocytes and fat body cells show the earliest molecular aging signatures, positioning them as key targets for intervention. Interestingly, neurons and glia age more slowly than other cell types. These findings suggest that different tissues require tailored strategies to slow aging and maintain function. In mice, single-cell atlases like PanSci and Tabula Muris Cinis have mapped over 300 cell types across multiple organs and life stages. These studies reveal that adipose, muscle, and epithelial cells are depleted early in adulthood, while immune cells expand later, contributing to chronic inflammation. The atlases also show that aging is not uniform. Some cell types, like adipose cells, age much faster than others. Machine learning models, or aging clocks, trained on these data can predict biological age and assess the impact of interventions, offering powerful tools for future research. A key insight from mouse aging atlases is the central role of the immune system in driving organismal aging. As mice age, naive T cells decline while cytotoxic and pro-inflammatory cells expand, leading to chronic low-grade inflammation, or inflammaging. These changes are linked to increased risk of infections, cancer, and age-related diseases. The atlases also show that aging microglia in the brain adopt gene expression patterns similar to those seen in Alzheimer's disease, highlighting the connection between normal aging and disease pathways. The diversity of aging across cell types demands customized therapies. Single-cell atlases reveal, for example, that in aged skin, keratinocytes drive inflammation while fibroblasts lose collagen production. This suggests using ligand-modified nanoparticles to deliver senolytics to keratinocytes, and viral vectors to enhance collagen synthesis in fibroblasts. Similarly, targeted delivery of IL-7 via exosomes can rejuvenate T-cell progenitors. By matching delivery systems to specific cell types and their unique aging drivers, future interventions can be both safer and more effective. Despite species differences, many core aging pathways are conserved from worms to humans. The decline of protein synthesis machinery, for example, is a hallmark of aging in both Drosophila and human blood cells. By comparing single-cell atlases across species, researchers can identify evolutionarily conserved targets for intervention. This cross-species approach strengthens the rationale for translating discoveries from model organisms into human therapies, focusing on pathways like mTOR and insulin IGF-1 signaling. Combining single-cell atlas data with findings from model organisms and advanced drug delivery technologies is transforming aging interventions. Dietary restriction, rapamycin, and metformin target conserved pathways, while single-cell atlases identify which cell types are most affected. This integration allows researchers to design interventions that are both broad and precise, engaging shared protective mechanisms while addressing cell type-specific vulnerabilities. As human single-cell atlases expand, they will guide the adaptation of effective interventions from animals to people. Aging is not a linear process. Molecular changes accelerate at certain ages, such as around 44 and 60 in humans, increasing disease risk. Single-cell data can help pinpoint when and where to intervene. For example, targeting lipid metabolism in endothelial cells around age 44 could reduce cardiovascular risk, while immune interventions at age 60 might rejuvenate T cells. By integrating temporal insights with cell-specific data, scientists can deliver precisely timed treatments to optimize outcomes. Advanced drug delivery systems are key to realizing the promise of precision longevity. Technologies like lipid nanoparticles, LNPs, GalNAc conjugates, and exosomes enable targeted delivery of therapies to specific tissues or cell types. For example, LNPs can deliver anti-inflammatory agents to adipocytes, while viral vectors provide long-term gene expression in fibroblasts. These platforms allow for multi-tissue targeting and tailored duration of effect, minimizing side effects and maximizing therapeutic impact. In summary, longevity research is moving rapidly toward precision, with single-cell atlases and advanced delivery technologies at the forefront. By understanding the unique aging trajectories of different cell types and tailoring interventions accordingly, we can develop therapies that are both safer and more effective. The integration of cellular resolution data with next-generation drug delivery holds the promise of truly individualized, cell-type, specific geroprotective interventions. As these tools expand to cover more human tissues, the dream of extending health span and reducing the burden of age-related disease is coming closer to reality.